Welcome back, ladies and gentlemen. So today we are going to pick up with Mendel's first hypothesis that he developed out of his pea plant experiments. And this was to simply understand that those alternative forms of characteristics he was seeing, alternative forms of a gene, he ended up calling them alleles. So when we talk about alleles, these are alternative forms for the same gene or for the same character. So alleles for a trait are located on corresponding positions on homologous chromosomes called their gene locus. So when we look in your genetic information, the information for that hair color gene or that eye color gene or that fur color gene or the purple flower gene, they all exist at the same location in a chromosome. So on chromosome 1, halfway down the chromosome, right? And that's important because when we are looking at meiosis, we talked about crossing over. We want to make sure that we're crossing over the same information as we're mixing up and making those recombinant chromosomes. Your cells also need to be able to understand where to go looking for this information when it comes time to expressing it, because you're now not only expressing the information on one chromosome, but also on the homologous chromosome. So if I'm going to go through and do transcription and translation of the eye color gene, I need to be able to understand that the eye color gene exists on this chromosome at this location, and I'm not only going to transcribe and translate it on the chromosome your mother gave you, but we're also going to do the exact same thing at the exact same time on the chromosome that your father gave you, so that the proteins that are created from both of those sets of information are then working together within your cells to then drive the physical phenotype, the physical trait that you end up seeing as either blue eyes or green eyes or brown eyes. So that's what we're looking at here, right? So each of those characteristics then exists on the chromosome from your mother at the same location it exists on the chromosome from your father. And then it's a question of what alleles, what variations on that genetic sequence did your mother and father pass on to you. That then translates into the proteins they produce, which ultimately translates into the physical expression. So in this case, for trait A, both the mother and father gave the dominant allele, which means the sequence should be more or less identical and the protein that results will be the same. If I look at the trait B, though, mom gave a recessive, dad gave a dominant, capital versus a lowercase. So the sequences here will be different. The proteins that they will produce will be slightly different. But if this is traditional Mendelian genetics, then the phenotype, the physical expression of those two proteins, will end up favoring this dominant protein. So remember, we talk about these homologous chromosomes. They're the matching chromosomes within an organism. When we look at a human set of traits, you've inherited one set, right, from your mother and one set from your father. So one chromosome 1 came from your mom, one chromosome 1 came from your dad, one chromosome 2 came from your mom, one chromosome 2 came from your dad, so on and so forth, right? And these all got separated out when we went through the process of meiosis to make those gametes. When those gametes came back together, egg plus sperm, we bring back together the two halves of the set to make a full set of 46 chromosomes in a human. Same thing works in other organisms, just that their chromosome number might be a little bit different. So ultimately, this is going to lead to this idea of chromosomal theory of inheritance. So these Mendelian genes have a specific location or loci on the chromosomes. The behavior of those chromosomes during meiosis, as well as during fertilization, ultimately account for the inherited patterns that we see. So the segregation of those chromosomes via meiosis, the independent assortment that happens, we could even throw in the idea of some crossing over and talking recombinant chromosomes. But all of that separation into those gametes and then bringing those gametes back together during fertilization accounts for the way that we see traits inherited. So genes we now know are found on the same location or gene locus on a homologous chromosome. And the cool thing is that by understanding this segregation, this independent assortment, the crossing over as well, we can start looking for the number of genes that get recombined in that process get mixed up from basically what your parents gave you to what you then give to your offspring. And that's how we're actually able to map where chromosomes exist, which is pretty cool. Actually being able to understand where the traits are not only on a single chromosome, but where they are within that chromosome, and create those chromosome maps, which is pretty neat. So again, it's this whole idea of taking these chromosomes and being able to go through meiosis and split them out so that you end up from these two characteristics on two independent chromosomes with four different gametes as a result. And again, this is going to be the driving understanding for being able to use things like Punnett squares to be able to predict the ratios of traits we should see in an offspring. And when we see those ratios exist, that's pretty exciting. When we see derivations from those expected results, we know something else is going on in terms of the way that inheritance is happening, and it's not following just this traditional Mendelian genetics. So again, there is the idea of a gene locus, right? They're existing at the same location on the homologous pairs of chromosomes that you got from your mother versus your father. Um, so when we go through transcription and translation, both of those sets of genes that are existing within this location are going to be transcribed and translated, and then the proteins that get associated with those sequences then intermix within the cells, and that's going to ultimately drive the physical expression that we end up seeing of that genetic information.